Welcome to Computational Biology. Today, we are diving into the fascinating world of drug discovery methods. Imagine trying to find a single, specific key to unlock a complex door, but you have billions of keys to choose from. Doing this in a physical laboratory takes years and costs billions of dollars. But what if we could use powerful supercomputers to simulate this entire process? In this lecture, we will explore how computational tools like molecular docking and virtual screening allow us to discover life-saving medicines in a fraction of the time. Let us get started. To begin our journey, let us understand the fundamental concept of how drugs work in the human body. Diseases are very often caused by malfunctioning proteins, which you can think of as highly complex three-dimensional locks. To stop a disease, we need to find a small chemical molecule, a ligand, that acts as the perfect key to fit into this protein and turn it off. In the past, scientists had to physically synthesize and test thousands of these keys in a lab. It was a slow, expensive process based largely on blind luck. Today, we use computers to simulate this matching process. Now that we understand the lock and key analogy, let us look at how computers actually solve this puzzle. This brings us to molecular docking. Molecular docking is a computational method used to predict exactly how a small molecule binds to a larger protein receptor. The software calculates the most stable three-dimensional orientation of the drug within the protein's binding site. It does this by simulating physical and chemical interactions, such as hydrogen bonding, electrostatic forces, and hydrophobic interactions. By evaluating these factors, the computer creates a mathematical model of a biological event before it ever happens in a test tube. This simulation process we just discussed relies on a fundamental thermodynamic equation. To determine if a drug will actually stick to a protein, computers calculate the change in Gibbs free energy. In this formula, delta G represents the change in Gibbs free energy, measured in kilocalories per mole. Delta H stands for the change in enthalpy, which is the heat energy released or absorbed when chemical bonds form. T is the absolute temperature of the environment in Kelvin. Finally, delta S represents the change in entropy, which measures the disorder of the system. For a drug to bind spontaneously, the final value of delta G must be negative. If enthalpy decreases or entropy increases, the binding becomes stronger. Before we move on, let us see this formula in action with a concrete example. Suppose we are testing a virtual compound designed to stop cancer cells. The docking software tells us the enthalpy change is negative 12.0 kilocalories per mole due to strong hydrogen bonds. The human body temperature is 310 Kelvin. The entropy change is negative 0.01 because the drug loses its freedom to move once bound. First, we multiply temperature by entropy to get negative 3.1. We subtract this from the enthalpy, giving us a final Gibbs free energy of negative 8.9 kilocalories per mole. Because this is negative, the drug will bind spontaneously. Now that we have used this formula, let us understand the thermodynamic reasoning behind why it works. In the physical world, systems naturally want to move towards a state of lower energy and higher stability. When a drug binds to a target, it forms chemical bonds. If these bonds are strong, like hydrogen bonds, heat energy is released. This makes the change in enthalpy, delta H, a negative value. A negative enthalpy heavily drives the entire equation towards a negative delta G. This mathematical proof of stability is exactly what the computer is evaluating when it scores a potential drug. Continuing our derivation, we must also consider the entropy, or disorder, of the system. When a free-floating drug gets stuck in a protein pocket, it loses its freedom of movement. This means the change in entropy, delta S, is usually negative. Subtracting a negative entropy term actually adds positive energy back into the system, which fights against binding. Therefore, for the final delta G to remain negative, the negative enthalpy from strong chemical bonds must be large enough to overcome this entropy penalty. When we mathematically prove that delta G is negative, we guarantee the drug-target interaction is stable enough for medical use. So we have proven that a negative delta G indicates stable binding. This brings us to a practical scenario, comparing two different drugs. Imagine we have drug A with a binding energy of negative 6.5 kilocalories per mole, and drug B with negative 9.2. Both target the exact same disease-causing protein. We need to choose the better candidate. Since negative 9.2 is a smaller, more negative number, drug B releases more free energy upon binding. This means it will hold onto the protein much more tightly than drug A. As a computational biologist, you would confidently select drug B. Let us visualize everything we have discussed about molecular docking so far. On the screen, you can see a schematic representing the lock and key mechanism. On the left, we have the receptor, which acts as the lock, and the ligand, which acts as the key. The arrow points to the docked complex on the right, where the ligand fits perfectly into the receptor's binding pocket. The text below reminds us that this spontaneous binding occurs because the change in Gibbs free energy is negative. This single technique forms the absolute backbone of modern computer-aided drug design. Now that we understand how to dock a single molecule, let us scale things up. Imagine you lost your keys on a massive sandy beach. Sifting by hand would take years, but a metal detector could find them in hours. Virtual screening is the computational metal detector for drug discovery. The chemical universe contains billions of possible drug-like molecules. It is physically and financially impossible to test all of them in a real lab. Instead, scientists use supercomputers to automatically test millions of different chemical compounds against a disease target, rapidly filtering out the useless ones. This massive scaling up leads us to the concept of the computational funnel. Virtual screening takes the detailed physics of molecular docking and applies it using automated pipelines and high-performance computing. The software docks every single molecule from a massive database into the target protein. It calculates a binding score for each one and then ranks them from best to worst. The top-ranked molecules, known as hits, are then selected for actual physical testing in a wet laboratory. This transforms drug discovery from a manual guessing game into a highly automated, data-driven science. To know if our massive computational funnel is actually working, we rely on a specific mathematical metric. This is the enrichment factor, denoted as E. It tells us how much better our computer model is compared to picking chemicals completely at random. In this formula, the numerator represents the success rate of our computer model. H sub S is the number of active drugs found, divided by N sub S, the total compounds the computer selected. The denominator is the random background success rate. H sub T is the total active drugs hidden in the database, divided by N sub T, the total database size. If E is greater than 1, our screening is successful. Let us apply this formula to a real-world screening campaign. Suppose a pharmaceutical company has a database of 100,000 compounds. Hidden inside are exactly 100 active drugs. We run our virtual screening program, and it selects the top 1,000 best-scoring compounds. When we physically test these 1,000 in the lab, we find 20 active drugs. First, we calculate our computer's success rate. 20 divided by 1,000 gives 0.02. Next, the random background rate. 100 divided by 100,000 gives 0.001. Dividing these rates gives us an enrichment factor of 20. Now that we have calculated an enrichment factor of 20, let us break down exactly what this proves about our computational methods. By setting up the ratio of the computer's success rate against the background success rate, we are mathematically evaluating probability. The background rate of 0.1% represents what would happen if we just blindly guessed which chemicals might cure the disease. The computer's rate of 2% represents the predictive power of our thermodynamic docking simulations. This chunk of our proof establishes that the computer is actively separating good drugs from useless ones. Concluding our proof, we look at the final value of the enrichment factor. Because 20 is significantly greater than 1, we have mathematically proven that our supercomputers are vastly superior to random trial and error. Specifically, our virtual screening program is 20 times better at finding active drugs than random guessing. This proves that the computational method is highly effective. By only synthesizing the top fraction of a percent of compounds with the highest mathematical probability of success, pharmaceutical companies save immense amounts of time and money. Let us look at a diagram that perfectly summarizes this entire high-throughput process. At the top, we have a massive cylinder representing the chemical database, containing millions of compounds. These flow down into a computer monitor, representing the high-throughput molecular docking phase. Below that, a large funnel physically filters the molecules, dropping only the top-ranked hits, which have a high enrichment factor, into a laboratory flask at the bottom for physical testing. This visual shows exactly how virtual screening acts as a massive computational funnel, revolutionizing the pharmaceutical industry. We have covered a tremendous amount of ground today, so let us review the most important ideas. First, molecular docking allows us to predict how a small drug molecule fits into a disease-causing protein, acting like a lock and key. Second, we use the Gibbs free energy equation to mathematically prove if this binding is stable and spontaneous. Finally, virtual screening scales this process up, acting as a computational funnel to search millions of compounds, and we measure its success using the enrichment factor. You now understand the core computational methods driving modern drug discovery.